You are about to step into a mystery where science meets the unknown. Imagine standing on the shore of an earth that does not yet know life. The sky is copper and violet. Lightning forks through volcanic haze. Waves crash against newborn continents. Hot springs bead and hiss like cauldrons. Meteorites hiss into steaming water and vanish. There are no eyes to watch, no cells to divide, only a planet of raw chemistry. Somewhere in this chaos, molecules begin to conspire. They link, fold, copy, and enclose themselves. And at some threshold, the world crosses from mere reaction to self-propagation. It is the most audacious transition in the history of matter. Lifeless molecules becoming living cells. And if you'd like to help me bring more of these mysteries to life, don't forget to like, comment, and subscribe. Tonight we follow the detectives of Abiogenesis as they hunt clues in ancient rocks, lightning bottles, deep-sea chimneys, and fragile protocells on a lab bench to ask, how did chemistry learn to want, to persist, to adapt, to remember? And what does that say about our place in the universe? The modern story begins with a thought from the 19th century. Charles Darwin once mused about a warm little pond where simple chemicals, energized by light or heat, could assemble into more complex forms. In the early 20th century, Alexander Oparin and J.B.S. Haldane translated that dream into a hypothesis. Without oxygen to rip things apart, early Earth's atmosphere and oceans might have been a chemical workshop that built the ingredients of life. In 1953, a graduate student sparked that workshop in glass. Stanley Miller filled a loop with simple gases thought to be primordial: methane, ammonia, hydrogen, water vapor, and zapped them with electricity to mimic lightning. Within days, the water turned pink. Analysis revealed amino acids, the building blocks of proteins. It was a cinematic moment. Bottles and sparks giving rise to biochemistry. Over the following decades, other experiments showed that hydrogen cyanide can condense into adenine, one of RNA's letters, that meteorites carry amino acids, sugars, and nucleobase precursors, that volcanic gases can yield a soup of organics under the right conditions. The Earth, it seemed, could cook. But cooking ingredients is not cooking life. Life requires 3 pillars: information, metabolism, and compartments. You need a way to store and copy instructions. You need a way to harvest and channel energy. And you need a boundary to keep the useful stuff in and the noise out. Modern cells use DNA for long-term information, RNA for messages and catalysts, proteins for most of the hard work, lipid membranes for walls, and a fantastically subtle energy economy where proton gradients turn molecular turbines. How could anything like this start without the machinery it depends on? One influential idea says, start with information. The RNA world hypothesis proposes that before DNA and proteins, there was an era when RNA did both jobs, carrying genetic information and catalyzing reactions. This is not fantasy. RNA in modern cells still catalyzes vital chemistry, including the heart of protein synthesis. In labs, ribozymes, RNA catalysts, can copy short stretches of RNA, ligate fragments, and perform tricks with surprising sophistication. Chemists have shown plausible ways to form nucleotides and link them into short strands under early Earth-like conditions, especially when minerals or cycles of wetting and drying drive the reactions. Fatty acids, which form readily from simple precursors, and even in space, self-assemble into vesicles, bubbles that bulge, divide, and reseal. Put the two together and you have a cartoon of a protocell. an RNA genome in a soft fatty acid container, capable of growth and occasional division. It is not life, but it is a tangle that begins to resemble it. Another school says, start with metabolism. Long before any molecule learned to copy itself, networks of reactions might have harvested geochemical gradients and ratcheted complexity upward. On the seafloor, alkaline hydrothermal vents, porous towers where mineral-rich, hydrogen-laden fluid seeps through rock into an acidic ocean, form natural batteries. Across tiny mineral membranes riddled with iron-sulfur clusters, a proton gradient hums. Some researchers argue that this environment could have powered the first carbon-fixing cycles, building molecules like acetyl groups, pyruvate, and beyond. Steps echoed today in ancient metabolic pathways. In this picture, chemistry finds self-maintaining loops first. Genetic polymers arrive later, parasitizing and then canalizing the flow. A third thread focuses on compartments. The lipid world. Because membranes arise so easily from amphiphiles, molecules with a water-loving head and a water-hating tail, perhaps early evolution belonged to populations of competing vesicles that grew by stealing lipids, divided under gentle shear, and captured catalysts by chance. Any internal chemistry that helped a vesicle grow, say by generating more amphiphiles from precursors, would spread. Information as a separate polymer could have emerged as a refinement rather than a starting point. Reality may have braided all three. In warm little ponds, shorelines that came and went with tides and seasons, cycles of drying and re-wetting concentrate monomers and drive the condensation reactions that water ordinarily punishes. Clays like montmorillonite can template nucleotides into growing strands. Freezing can also help. In ice, salts and organics are excluded from the forming crystals and become concentrated in briny channels where reactions proceed in a cold, protected environment. UV light, destructive at full blast, also powers photochemistry that builds sugars and nucleotides from simple gases. Meanwhile, meteorites and interplanetary dust drizzled organics onto the planet for hundreds of millions of years, adding richness to the mix. The chemistry is plausible, even playful, and yet the hardest problems sit like boulders in the stream. Here is why abiogenesis is difficult. First, water is both friend and foe. It transports and moderates, but it also breaks the very bonds you want to form. That is why cycles, wet to bring molecules together, dry to drive condensation, are so attractive. Second, yields are fickle. Organic precursors tend to form messy tars unless carefully guided, a phenomenon some call the asphalt problem. Third, ribose, the sugar in RNA, is fragile. The classic formose reaction that makes sugars from formaldehyde produces a jumble, unless stabilized by borate minerals or other helpers. Fourth, phosphate, the backbone of nucleotides, is not easily mobilized, and activating it for polymerization without enzymes is non-trivial. Fifth, chirality. Life uses left-handed amino acids and right-handed sugars. Prebiotic syntheses make both hands unless some bias selects one over the other. Finally, copying. Even if you have an RNA polymer forming strands, can it copy itself with enough fidelity to avoid an error catastrophe? Can a protocell keep its genome intact through cycles of growth and division? Each question hides layers, and above them all looms Earth's deep past. We do not have rocks from the first few 100 million years to settle the argument. Only isotopic whispers in ancient minerals and the habits of microbes, billions of years younger. Still, progress accumulates. Chemists have coaxed nucleotides to form under plausible conditions, starting from hydrogen cyanide and derivatives, avoiding the messy formose step in yielding the right sugar-base combinations. Fatty acid vesicles grow by absorbing simple lipids and by making more from oily precursors. They can divide under gentle flows and can be stabilized by salts thought to be present in early seas. Ribozyme engineers have evolved RNA that can add nucleotides to templates, and with help from mineral surfaces and cycles, extend strands many dozens of letters long. Simpler genetic polymers than RNA, tNA, pNA, and others form more easily and could have served as stepping stones, later handing information off to RNA. Mineral microcavities and alkaline vents act like natural cells, with catalytic iron-sulfur cofactors embedded in their walls. They generate hydrogen and organic molecules and maintain proton gradients, nature's first batteries, without any proteins at all. Wet-dry cycles in little rock pools produce multi-layered films that, upon rehydration, peel into vesicles carrying entrapped polymers. A primitive version of packaging. None of these results is definitive. All of them are tantalizing. At this point, the field branches into major hypotheses with pros and cons. The RNA world has the advantage of continuity. RNA still sits at life's core, but struggles with how to get ribozymes robust enough to copy long genomes without proteins, and how to make and activate nucleotides in messy environments. Metabolism-first models thrive on geochemical realism. There were vents, and they do make gradients, but must show how specific, self-amplifying networks avoid dissolving into chemistry's noise and how they entrain information polymers later. Lipid-first scenarios demonstrate easy compartments, but must bridge to heritable complexity. Co-evolution models try to soften those divides. Perhaps short RNAs first aided simple metabolism, improving lipid growth. Then better catalysts emerged inside vesicles. Eventually, true replication and selection took hold. In some tellings, life began not in the ocean, but at the edges. Shorelines and hot springs where concentration, cycling, and diverse chemistries coexist. Others insist on the deep, stable power of vents, continuous energy and protection from surface cataclysms. A minority argues for ice, cold concentration, less hydrolysis, gentler conditions for fragile polymers. The truth may be many truths. Multiple locales seeding multiple chemistries, with only one lineage lucky, and robust enough to take over the planet. There are respectable, if controversial, side quests. Panspermia suggests life or its building blocks arrive from space, not to dodge abiogenesis, but to move it. Meteorites like Murchison carry amino acids, sugars, and nucleobases. Comets harbor complex organics. Mars once had lakes and hydrothermal systems, and may have offered friendlier early surfaces than Earth's restless crust. It is possible the spark lit there and hopped here via impact-launched rocks. That shifts the locale, not the problem. But it would mean life's emergence is not uniquely terrestrial. Some wonder about a shadow biosphere on Earth. Organisms so chemically different we have missed them. Those searches have yet to reveal truly alien lineages among us. Others imagine early life based on alternative backbones, with amino acids or hydrocarbons as scaffolds for heredity before RNA. Mainstream science treats these with open curiosity and hard standards. Show us a pathway. Show us selective advantage. Show us continuity with what we see today, or a reason for its absence. We can peer back through evolution's lens as well. Comparative genomics sketches the last universal common ancestor, LUCA, not as the first life, but as the root of modern cells. Luca seems to have had a translation system, a genetic code, ribozymes, and a metabolism tuned to gases like hydrogen and carbon dioxide, with enzymes enriched in iron-sulfur cofactors. That is a whisper of environments where metals and redox chemistry mattered, vents again, or mineral-rich hot springs. The membrane story is more nuanced. Bacteria and archaea use different lipids and membrane assembly routes, hinting that membranes diversified after LUCA, or that earlier cells swapped parts. Either way, the universal features, ribosomes, the code's grammar, ATP's role, tell us that once heredity and catalysis joined, the particulars locked in. The ladder of life may have many rungs, but the last few were decisive. Beyond Earth, the implications are enormous. If life's origin required rare coincidences and exquisite conditions, we might be alone across light years. If instead it was a near inevitable outcome of planets with water, energy, and time, the cosmos could be teeming with metabolisms. Our probes sniff plumes at Enceladus and Europa for organics and hydrogen. Rovers grind Martian rocks in ancient lakebeds. Telescopes read the colors of exoplanet atmospheres for gases out of equilibrium. Prebiotic chemistry is not just a story about our past. It is a script for where to look next. Alkaline vents on ocean worlds, UV-based shorelines on M-dwarf planets, hydrothermal pools under Titan skies. Each is a stage where the same physics, gradients, cycles, catalysis, selection, might play variations on the theme. And then there is the philosophical cargo. Life's origin is a boundary question. When does a pattern deserve the word alive? Is life a property of matter arranged a certain way, or a process? Information maintained far from equilibrium by constant work. Abiogenesis suggests an answer. Life is what happens when chemistry acquires memory and motive, when reactions begin to select for their own persistence. That may feel deflationary. No breath from outside, only a long chemical dusk giving way to dawn. Or it may feel more miraculous. Atoms writing a story about themselves that never has to end if they keep turning pages. Either way, the question is not whether life began by natural means. It must have. But how many roads there were, how long they were, and whether some were short enough to be common. We should also admit the human angle. We love tidy tales, lightning in a bottle, a single warm pond, a final triumphant experiment. The truth is patient and plural. It lives in small advances, a ribozyme that adds a few more letters, a lipid vesicle that can withstand salt, a mineral that biases one hand of a molecule, a network that closes a loop. It lives in arguments about early atmospheres and tectonics, in debates about whether UV light was friend or foe, in revisions to classic experiments when we learned the gases were wrong. It lives in cross-disciplinary collisions where geologists, chemists, biologists, and astronomers learn each other's languages. The origin of life is not a finish line we will break through to applause. It is a map that gets clearer, then foggier, then clearer again, with landmarks accumulating until the landscape stops feeling alien. So where do we stand? We can plausibly make many of life's parts under early Earth conditions. Amino acids, fatty acids, nucleotides or their precursors. And we can coax those parts into protocells that grow, divide, and perform simple chemistries. We have models in which energy gradients are available and persistent. We know minerals that catalyze key steps. We have experimental cycles that take unpromising mixtures and push them uphill. We do not yet have a closed loop, a protocell that can copy a polymer long enough to encode its own growth and division, improving through selection over many generations without modern enzymes. The gap is narrowing. The challenge remains beautiful. As our camera pulls back from the primordial shore, try to hear what the planet was saying before any ears existed. Keep gradients. Build scaffolds. Hold on to what works. Discard what does not. Remember, from those imperatives, a world of cells, tissues, and minds unfolded. If matter can do that once, maybe it can do it often. If it did it here, maybe it has done it in pools beneath alien skies. And if the path was long and narrow, then our little thread of chemistry is all the more precious. Either way, the question that once haunted a philosopher by a seaside has grown into a program of experiments across continents and worlds. We started with a storm and a silence. We end with a whisper and a wager. That the boundary between life and non-life is not a wall, but a shoreline. Cross where information, energy, and compartments embrace. I'll leave you with this. If you were going to search for life's spark beyond Earth, would you choose the darkness and power of a vent, the rhythm of a drying pool, the clarity of ice, or the chemistry of a different planet entirely? And why?